Right, in the second half of chapter 3, we're going to continue talking through the 4 stages of metastasis. So, in the first half, we discussed primarily angiogenesis, the ability of tumors to recruit their own blood supply. And of course, once that blood supply is there in proximity to the tumor, then it makes it much easier for cancer cells to invade those vessels and be distributed systemically. So if we think about this process of invasion, there's actually a few things that will have to happen. And really the first step would be that the cells will have to detach themselves from that primary tumor and move to the blood vessels. And so this kind of begs the question, okay, well, what is it that's normally holding those cells together? And of course, there's a number of different molecules involved, but one of the key players seems to be this protein called E-cadherin, which you can see illustrated here, which I think I mentioned in previous chapters, almost like literally like Velcro on the surface of these cells. So kind of a hook and eye type connection where you have these T-shaped E-cadherin molecules that will interlock at the cell surface and help maintain the integrity and the adhesion of those tumor cells to each other. So what we see as these cancer cells evolve is they will downregulate the amount of E-cadherin protein that they're producing, so it becomes easier for those cells to detach. And we can basically mimic that in vivo by treating with antibodies that will block E-cadherin protein that has also been shown to enhance cancer spread. So we know for sure that an abundance of E-cadherin would typically reduce the ability of cancer cells to metastasize. So we'll see those cells detach, and then once they're detached, of course, they have to be able to move, even though it's likely a relatively short distance, they have to be able to move to the vessels. And to do that, they're going to be moving through what is probably primarily a matrix of the extracellular material, extracellular matrix. And if we think of cell movement, this is primarily going to be activated by inducing reorganization of microfilaments or the actin cytoskeleton inside of a cell. So you can see here these actin fibers, and then there's a host of other proteins that are involved with kind of cross-linking um, and mediating the interaction of these filaments with each other. And then keep in mind you have the integrin proteins, right, that have a portion inside the cell interacting with the microfilaments, and then a portion outside the cell interacting with the extracellular matrix. And in a nutshell, what will happen is those microfilaments, those actin filaments, will be dramatically reorganized just under the plasma membrane at one end of the cell. And that reorganization, and really it's going to be a polymerization and a growth of those microfilaments, will push forward one end of the plasma membrane and form what's called a lamellipodium, which translates as a false foot, so it's sticking its foot forward essentially. And then at the same time that you have that happening at one end of the cell, at the other end, you have myosin motor proteins that are also interacting with the actin cytoskeleton, causing a contraction. So it's essentially almost like a muscle contraction at the opposite end of the cell, and that's going to force the cytoplasm and the cell contents off in the direction of that lamellipodia. So those two things happening repeatedly and in concert with each other are in this case going to force this cell to move off to the right. And you'll also notice here these little red lines you may be able to see here at the bottom are representing some of those integrin proteins that are going to mediate the interaction between the cell and the extracellular matrix, represented in yellow here. So they're basically providing that traction, sort of gripping onto the extracellular matrix and allowing the cell to move forward. Here you can see a cluster of cancer cells that are in the process of moving. So they've got the cell-to-cell adhesion protein labeled with red. So you can sort of see the outline of some of these cells in red here. So you've got this central cluster of cells. Much of the blue here is going to be extracellular matrix, and the green is labeling the integrin protein. And you can see that integrin is focused at the front end of the cluster of cells in this case, right, where it's interacting primarily with the extracellular matrix. So you can think of this as this cluster of cells basically grabbing hold of the extracellular matrix and then pulling itself forward using those cytoskeletal rearrangements that we just discussed. So we detach, we activate motility so the cells can move, and then as we just saw in that image, right, the cells are having to move through extracellular matrix. So to sort of clear a path, they're typically going to start producing proteases, which are protein degrading enzymes that are going to allow it to not only penetrate what's called the basal lamina, the layer, sort of the outer layer of those vessels, but also degrade proteins that may be part of the extracellular matrix, and as I said, clear that path, so to speak. And so a lot of this activity is tied in with this protein called plasminogen activator, which gets produced. And what plasminogen activator will do, as its name implies, is it will activate plasminogen, which is typically almost always present in high concentrations in most tissues and fluids in the human body. But plasminogen is inactive, doesn't really have any biological activity until plasminogen activator comes along, converts plasminogen into plasmin, which is then biologically active as a protease, can degrade extracellular matrix. Plasmin will also convert these matrix metalloproteinase precursors into active MMPs, so basically another second layer of proteases, which will then further degrade extracellular matrix and help chew through that basal lamina. And the last aspect I wanted to mention of this, something that's a little bit newer and isn't that well understood, but it's what are called these nanotubes. And we know cancer cells can produce these nanotubes. You see a cancer cell highlighted with a star here and an X down here. This would be a small vessel, like a capillary, and you can see the cancer cells attached and has sent out these essentially tendrils. Those are the nanotubes for attachment. You can see the same thing here, right? This cell is actually in the process of penetrating into this vessel. But you can see some of these nanotubes extending and aiding in that attachment and adhesion process. And interestingly, these nanotubes also seem to be involved with communication. So here we see two cancer cells that are actually connected to each other through what are called tunneling nanotubes. And you can see material here moving through those tubes from one cancer cell to the next. So it's been shown that small signaling molecules and even mitochondria may be able to move directly from cell to cell through these kind of tubes. So the significance of that is not entirely clear, but obviously that kind of connection would aid in the ability of cancer cells to communicate with each other and maybe coordinate their activity and movement. All right, so cancer cells will detach from the tumor, move through the extracellular matrix, and invade the circulatory system. And then the third stage of metastasis is going to be their transport through the bloodstream to distant parts of the body. And I should start this by saying most cells are not going to survive the trip through the bloodstream. So, we talked about the fact that cancer cells are kind of unique in that they can typically divide and thrive in suspension. They don't necessarily have to be attached to a substrate. But that being said, there's still lots of obstacles, immune cells, phagocytic cells that are going to make that journey difficult even for a cancer cell. So it's been estimated that even a small tumor can release potentially millions of cancer cells into the bloodstream every day. That's a pretty terrifying number. Um, but this research also shows that maybe only 1 in 1,000 of those cancer cells are actually surviving that trip. But still, if you have, a million released every day, even if only 1 in 1,000 make it, that's still a lot of cancer cells being distributed through the body. Um, and I also wanted to point out that, instead of going directly into blood vessels or capillaries, a lot of times cancer cells may initially penetrate lymphatic vessels. And so if you're not as familiar with the lymphatic system, I typically think of this as sort of a shadow circulatory system. And it is a very dense network um, of lymphatic vessels and lymph nodes, right, that basically spreads throughout the whole body. So you can see this illustrated here on the right. Um, and there's a couple of key jobs of the lymphatic system. One of its main roles is to help collect fluid that is being lost through capillaries, diffusing out of capillaries into surrounding tissues, basically recollecting some of that fluid and recycling it back into the bloodstream. And then another key role of the lymphatic system is an immune role. So you do have immune cells that tend to be concentrated primarily in the lymph nodes, right, where you have filtering going on and those immune cells attacking what are identified as foreign substances that may be moving through the lymph node. But the bottom line is this lymphatic system, right, provides a nice uh, pathway for cancer cells to move as well through the body. And so, you may be familiar with the idea that a lot of times cancer cells will actually collect at lymph nodes, and typically, if you're having a surgery to have a tumor removed, the surgeons are going to try to look at biopsy, maybe even remove lymph nodes in the area of that primary tumor to look and see if there's any evidence of cancer cells having spread to those nearby lymph nodes, because they will almost always be collected and concentrated at those nodes. So if you do see cancer cells in the lymph nodes, right, that's a good indication that, yeah, this primary tumor was already metastasizing and may suggest that you may find cancer cells in other parts of the body as well. So some of that movement may be through the lymphatic system. And another thing that researchers have determined over the years is that cancer cells can actually evolve to become better at metastasizing over time. So some simple experiments like this in mice really illustrate that point. And what they did in this particular experiment is they were injecting, I believe, human melanoma cells into the mouse, and then waiting for those cancer cells to metastasize to the lungs. They would then remove some of the cancer cells from those metastases, inject them into the second round of mice, wait for those to metastasize, collect the metastases, and repeat, so on. So they would go through basically 10 cycles of this kind of injection metastasis collection. And what they would find is at the end of those 10 cycles is the injected mice were forming many, many more metastases in their lungs, suggesting that that population of cancer cells was much more efficient at metastasizing than the original melanoma cells that we started with. So we can definitely see tumors, cancer cells changing over time and becoming better and better at metastasizing. So again, another reason you would want to try to identify and remove tumors as early as possible. And so another thing they did in these studies was look to see, okay, well, what actually changed in these cells? Why are they better at metastasizing? And they did some sort of widespread gene expression analysis, basically looking to see which genes have had their expression changed, are being expressed more or less, transcribed more or less, compared to the cells in the beginning of the study. And what they found was there seemed to be about 17 sort of key genes whose expression patterns were being changed, and that seemed to tie in with metastasis. And certainly those genes are going to be things like we just talked about, right? So, um, the changes in E-cadherin, changes in proteins that might help rearrange the actin cytoskeleton for movement, uh, changes in genes like plasminogen activator that might help with digestion of the extracellular matrix. But those genes are also going to provide some nice candidates for further study and potentially even for new cancer treatments or drug development. And so then the fourth stage of metastasis is going to be exit from the bloodstream and establishment of new tumors. Of course, that seems pretty straightforward, but I did want to go through this process, which is called extravasation. And this is going to begin when a tumor cell is going to get lodged in a small capillary. So the cancer cell gets stuck in the capillary here. Platelets will accumulate, right? So it's basically a clot for all intents and purposes. Um, the body will eventually send proteases through the bloodstream to try to dissolve that clot, but of course they're not going to be successful in dissolving the cancer cell. But while this is happening, that cancer cell will typically push aside the endothelial cell layer that's shown in green here and attach itself directly to the basement membrane. And once it's attached to that substrate, then it will begin to divide and proliferate pretty quickly. And what will happen is it'll just rupture through that capillary, and then those cells are free to start either moving or dividing out into the surrounding tissue. So, it looks like there's a lot going on here, but all this is going to happen pretty quickly within just a few days. And I also want to point out that there's actually a lot going on in these simple steps that we just walked through. So I think all the dirty details are a bit beyond our scope for this particular class, but this is basically a more detailed view of that same kind of image. So your tumor cell here lodged in the capillary. And I just wanted to point out that there's all kinds of interactions happening here, right? So you've got interactions between the tumor cell and those platelets, um, between the cancer cell and immune cells that may be circulating, macrophages, neutrophils, between the cancer cell and these endothelial cells, right? So there's all kinds of signaling molecules and signaling pathways that would be activated by these various interactions. There's also a fair amount of evidence just from studying the trends in metastasis over the years that in many cases where cancers metastasize may be largely dependent just on the direction of blood flow in the body. So, for example, if you had a gastrointestinal, stomach, or colon cancer, it will very often metastasize to the liver, just due to the simple fact that that's where the blood flow is carrying those cancer cells first from the gastrointestinal tract. Um, Lung cancers will typically be distributed widely throughout the body when they metastasize because the blood flow is going from the lungs, right? Taking that oxygenated blood to basically all parts of the body. So in some cases, you may be able to predict uh, metastasis just based off of blood flow. But that being said, there are some interesting examples where cancers tend to spread to tissues that you wouldn't predict based off of blood flow. So, for example, prostate cancer often metastasizes to bone. And these kind of unique situations have led to what's called the seed and soil hypothesis. The idea being that the cancer cell is the seed, and for a seed to grow, you need the appropriate soil, right, with the appropriate nutrients, the appropriate amount of water. So in this case, the soil would be the specific tissue that is going to have all the appropriate signals and if you want to think of them as nutrients that will allow that cancer cell to grow the best, right? So for whatever reason, uh, prostate cancer cells seem to grow very well in bone tissue, right? It's the best soil for that particular seed. And there has been quite a bit of work going on in recent years looking at what's called the tumor microenvironment. So these would be the factors in these tissues that are helping to propagate cell growth. And in fact, in some cases, there may be, very specific growth factors or even signaling molecules that may help attract these cancer cells to these specific tissues or specific organs. And so I wanted to introduce this idea of chemokines, which are these very potent uh, signaling molecules, chemical signaling molecules that interact with what are called chemokine receptors. Those are the true like receptor ligand type interaction. And there have been some interesting examples. So certain types of breast cancer will produce a specific chemokine receptor, and the ligand for that, um, you'll find in lung or in lymph nodes. So it's almost like those breast cancer cells are being directly attracted to these individual tissues by these uh, due to this signal ligand type interaction. So just to summarize what we've gone over so far, so we have the formation of our primary tumor, right? Angiogenesis, bringing a vascular blood supply into that tumor, allowing it to start growing more rapidly. We get detachment of the cancer cells, localized invasion, um, intravasation, which is the entrance into the bloodstream, spread through the circulatory system, right? Um, Getting stuck in those capillaries, those microvessels in various organs throughout the body, that process of extravasation that we talked about, leaving the bloodstream, and then you'll get the formation of what we call small metastases throughout the body. And then if those micrometastases are able to recruit their own blood supply through angiogenesis, right, they may evolve into metastatic tumors themselves. And we have the whole process repeat at that point. And before we end here, I also just wanted to remind you that there's all kinds of interactions happening throughout this process of metastasis between the cancer cells and other cell types and other molecules throughout the body. So you've got the interaction of cancer cells with endothelial cells in terms of angiogenesis and in terms of being able to invade the circulatory system. You, of course, have almost constant interaction with immune cells, especially within the circulatory system or the lymphatic system. Um, Interaction with other normal cells in the tissues wherever those cancer cells happen to be growing. And then you also have interaction with the molecules and proteins in the extracellular matrix and those molecules and cells making up the basal lamina as well. So it gets quite complex if you think of it in terms of big picture and all these interactions that are having to happen. And lastly, I just wanted to reemphasize the fact that because metastasis is so important and is so deadly, um, it is a very attractive drug target, right? So anything that could be done to limit metastasis might reduce the deadliness, the severity of particular cancers. So, we talked about angiogenesis inhibitors, and there's a bunch of those that have been developed. But just to give you another example, we talked about those matrix metalloproteinases. Um, so, anything that might inhibit those, right, might reduce the ability of cancer cells to be able to move, to be able to metastasize. So I think we're really just kind of at the beginning of this ongoing search for genes, for molecules that are important for metastasis, And the more we can understand this process, right, the better we're going to understand how that process might be able to be blocked and inhibited. And just to remind you, I am going to try to hopefully have you watch this Cancer Warrior video that will delve into sort of the research story behind angiogenesis. So you can look for that in the near future.